Well, hello and welcome everyone to this June webinar, which is a joint project of the European Society for Toxicology in Vitro and the American Society for Cellular and Computational Toxicology. I'm Christy Sullivan and I'm the secretary of the ASCCT. Both of our organizations host these monthly webinars related to aspects of in vitro and computational toxicology, as well as pursue other activities to promote computational and in vitro toxicology fields and activities. We also, both of our organizations, offer training and education opportunities and aim to support early career scientists. So feel free to join or check out our websites to learn more about our organizations. Just a quick housekeeping, this webinar is recorded and the recording will be posted on the AICCT website a few days from now, and any slides that we can make available will be there. Also, the lines are muted. You can ask questions of the presenters using the Q&A feature at the toolbar on the bottom of your screen. So just type your question in and we will read it out after the presenter is finished. And we won't have, we'll have Q&A after each presentation. So we'll have the first speaker, then Q&A, and then the second speaker, and then Q&A. So definitely feel free to type your questions as they occur to you. I just wanted to mention a couple of brief items. The first is that ESTIV has a couple of activities coming up. They have their annual Congress next year in Barcelona, and you can visit their website to learn more about that. And there's also an applied toxicology course at the Luxembourg Institute of Science and Technology that's going to be in October of this year. ASCCT also has its annual meeting coming up. This is going to be a virtual meeting in October, so all are welcome. There's a few themes for the sessions, generally converging around practical applications of new tools in toxicology. We have a great lineup of plenary speakers for those sessions. We also choose oral talks from submitted abstracts as well as posters from submitted abstracts. And there are a lot of activities related to engagement between young and early career scientists and more established toxicologists. Lots of engagement opportunities, so feel free to join us. And July 23rd is the abstract deadline, so I hope you'll think about doing that, about submitting. The next webinar in this series is going to be July 21st, and the speakers are Marco Travaglio from University of Cambridge and Maria João Amorim from University Instituto Gulbenkian de Ciência in Portugal. They are going to be talking about different initiatives related to COVID-19. And you can register now for that webinar at our website. You can also see an archive of all of our past webinars. And really quickly, I wanted to mention, in case you are interested, we heard that the Birth Defects Research Journal is calling for papers for a special issue on stem cells for research to study birth defects and disease. And manuscripts are due December 15th, and you are advised to contact the editors of that journal to get more information. And so finally, I will introduce our presenters. I'm going to introduce both of them now, and then I'll let them take the floor. First is Dr. Catherine Verfaillie from Leuven University, and she's going to be talking about multicellular 3D liver models based on hiPSC-derived liver cells. Dr. Verfaillie received her medical degree from the University of Leuven and then went to the University of Minnesota for a postdoctoral fellowship and continued in a number of positions there, including professor, associate and full professor. In 2001, she became the director of the University of Minnesota's Stem Cell Institute and moved to the Stem Cell Institute at Leuven University in 2006. She has a longstanding career in stem cell biology, initially focusing on normal hematopoietic stem cells and leukemic stem cells, as well as pluripotent stem cells. The current research of the Verfaillie lab is focused on understanding what regulates self-renewal and de-differentiation of adult as well as embryonic or induced pluripotent stem cells, and creating stem cell-based models to study liver and central nervous system disorders. And then our second presenter will be Anne-Marie Vinggard from National Food Institute at the Technical University of Denmark. She's going to be talking about novel human pluripotent stem cell-based assays to predict developmental toxicity. And Anne-Marie Vinggard is a full professor of molecular toxicology at the National Food Institute, as I said. She conducts research with the overall aim of improving chemical food safety and the safety of consumer products. As an author of more than 120 scientific articles, she has particularly been engaged in understanding the long-term health impacts of early life exposure to environmental chemicals, with a special focus on chemicals with anti-androgenic action and mixture effects of chemicals. So with that, I will stop sharing and give the floor to Professor Verfaillie.
Can you see my screen or not?
We can see your screen, but it's not in. Yeah, there we go.
Okay. So this should be okay now, right?
Yep, looks good. Please go ahead. Thank you.
Good afternoon, everybody. First of all, I would like to thank the organizers for the invitation to give you some insights on using liver models based on pluripotent stem cells, derived hepatocytes, but also non-parenchymal cells, with the ultimate aim for toxicology studies. I would also like to thank you for the very kind introduction. As I said, I will be discussing today how you can create less complex and more complex liver models from pluripotent stem cells, including hepatocytes, but also non-parenchymal cells, for disease modeling of the liver, but also for toxicology studies in the liver. So why stem cells rather than, or in addition to, animal models? Well, obviously disease modeling in animals has contributed greatly to many insights we have in diseases. However, they can only model specific genetic diseases and to a much lesser degree sporadic diseases, including a number of liver disorders. For drug toxicity, animal models, in particular mice and rats, are used very extensively to study chemical and drug toxicity, but significant interspecies differences exist, and so they are also not fully predictive of drug toxicity. So then you could say, well, why don't you use thermally differentiated cells, and why don't you use hepatocytes, which you obviously can? But they are not always easily obtained. So, currently hepatocytes are obtained from cadaveric livers usually that are not good enough for transplantation. So in many cases, they're not perfectly healthy hepatocytes. Such cells are relatively difficult to genetically manipulate. It's very well known for hepatocytes, but also non-parenchymal cells of the liver, that they very quickly lose their phenotype if you put them in culture. Plus, there's a problem of high interdonor variability, which is interesting because it may represent the variation in the population. On the other hand, it also makes it a bit more complicated to interpret the assays that one sets up. Just an example from a paper from Godoy et al., where they showed that if you start with fresh human hepatocytes and you look over time in culture, what happens to them is that you can see that there is de-differentiation if you culture the cells. In 2D cultures, it is a bit less pronounced if you do a sandwich culture, but still there is de-differentiation, which is still happening for hepatocytes in vitro. And this really also means that you lose CYP for 50 functions and a number of other features that you need to do toxicology. Then if you culture primary human hepatocytes in spheroids, either on their own, as shown here on the left-hand side, and this is a paper from the Karolinska group, they can be maintained, and you can see that you maintain CYP3A4 and albumin expression if you add non-parenchymal cells, and hereby I mean all cells in the liver that aren't hepatocytes. So these would be Kupffer cells, these would be immune cells, these would be stellate cells and endothelial cells. You can also maintain the system in vitro. And now this model obviously might represent the liver quite okay. The problem, however, that investigators see is that if you take primary hepatocytes from one donor and mix them with non-parenchymal cells from two different donors, that the non-parenchymal cells, just like the primary hepatocytes, are highly variable, and that your answers, for instance, this here is a response to TGF-beta, is different. And the same is true for if you try to make a model for NASH and you add free fatty acids, you can see that depending on the primary hepatocyte source you use and the non-parenchymal cell population you use, you get variable responses. As I mentioned, this may not be so bad because it might represent different genetics in humans. But obviously, if you want to drug discovery or you want to do toxicology, it is not so simple if your baseline is highly variable. So therefore, you can use stem cells. And so the two possible stem cell populations would be to take adult stem cells or to take pluripotent stem cells. And then the question is, how do you use these stem cells to create a model of disease? So you can do monolayer cell culture, so one cell type at a time in classical 2D cultures, or you could use what we call spheroids or organoids, or you could make tissue explants, but obviously these would not be stem cell derived anymore. And if you want to do it even more complex, you can look at interplay between different of these models in multiplexed models. The group from Utrecht obviously has spearheaded organoid cultures. And so what they show is that you can indeed isolate stem cells from the liver, expand them, and this in this particular case was done in spinner flasks to make very large numbers of these cells, and then allow them to differentiate either spontaneously or by putting them on a disk of extracellular matrix of the liver. And then you can see that you get cells that start expressing a number of markers of hepatocytes. They also demonstrate that these cells start expressing cytochrome P450 gene expression and proteins, and also become functional. If you then do co-differentiation of, we need to go back one slide, so my excuses. So one of the problems here is that you have endoderm, but you don't have the mesodermal component. So if you really try to make a liver model, you don't have endothelial cells, you don't have hematopoietic cells, and you don't have stellate cells. So investigators are now trying to do co-cultures of the different cell components together with these organoids, but this is still in its infancy. Therefore, a number of teams are now looking into the use of ES cells or human-induced pluripotent stem cells. And one very good example is the group in Japan that actually makes liver organoids by co-differentiating multiple cell types from iPS. So essentially, they take induced pluripotent stem cells or embryonic stem cells, they make them into small organoids or small spheroids, and let them spontaneously differentiate into first mesendoderm, endoderm, and then in something that contains a number of different cells from the liver. So these cultures can contain a lot of hepatocyte-like cells, but also biliary-like cells, and a small fraction of Kupffer cells, because these are theoretically not generated by the liver in vivo, but are generated in a different organ. And there is a population of cells that have some stellate cell-like features. If they do single-cell RNA sequencing of this construct, what they see is that 60% of the cell have a hepatocyte-like phenotype, 5% have a biliary cell-like phenotype, 31% are more fibroblastic, and a very small population, the green cells here, are hematopoietic or Kupffer cell-like. If they then use this culture to have a look at the effect of lipids, they show that these organoids can actually become lipid-laden. And that, as you see in NASH, but also in other diseases like toxicity that causes lipidosis of the liver, that you get increased expression of IL-6, and you get increased presence of extracellular matrix, here shown by trichrome staining. And in a most recent paper in 2021, they started to look at this organoid system to look at toxicology. And I'm not going to go in detail, but essentially the system allowed them to identify cholestasis-inducing drugs and mitotox-inducing chemicals. Because in the previous model, you have a relatively infrequent number of macrophages and Kupffer cells, you have no endothelial cells. The alternative possibility is what we have looked at, is whether you could take induced pluripotent stem cells and differentiate them to early precursors for different cell populations, and then allow these to self-assemble into spheroids, probably not organoids, but spheroids, and then let them co-differentiate, but in a more defined ratio between the different cell types. And so we indeed developed a huge number of culture systems to first take iPS and get them to go to the hepatocyte lineage, iPS to let them go to stellate cell-like cells, Kupffer cells, I wouldn't call them Kupffer cells, but I would say macrophages, and then some cells with endothelial cells or even some features of liver sinusoidal endothelial cells. And then see if we can then combine these in, and we used custom-made hydrogel that mimics the liver extracellular matrix liver stiffness in optimized medium, which ultimately is not yet anatomically correct. So to make hepatocyte progeny from iPS, some of you who are familiar with the field know that there is many differentiation protocols, and they all generate cells with some features of hepatocytes. However, if you look at the cells that you can make in 2D, in green are transcriptome studies of primary hepatocytes, and in red are transcriptome studies of hepatocyte-like cells, as we call them, that are published, and in blue, the cells that we made a few years back. So these seem to be still quite different, and that's also very clear if you look at the function of these hepatocyte-like cells. They are not quite as good as primary hepatocytes. The transcriptome studies demonstrate to us that actually there are a number of different major differences between primary hepatocytes and hepatocyte-like cells. And those included cell respiration, in the sense that the cells, that hepatocyte-like cells actually do not use oxidative phosphorylation, but use glycolysis as energy, as ATP production system. But interestingly, we saw that in this big turquoise cluster, this was actually associated with also drug biotransformation enzymes. And so we set about trying to figure out if we could fix this turquoise cluster and actually make the cell switch from glycolysis to oxidative phosphorylation, which obviously hepatocytes do, and if this would actually also cause the drug biotransformation and therefore toxicology sensitivity to improve. So as I mentioned, HLCs have poor CYP3A4 function, which is shown here by BFC metabolization. It's reflected in their inability to metabolize APAP. And what we showed is that the cells maintain glucose consumption, but do not, like primary hepatocytes, produce glucose, and actually have very poor respiration. Here in green are primary hepatocytes, and in the bottom, you can see in blue hepatocyte-like cells on day 20, which just like iPSC, actually use glycolysis, but not oxidative phosphorylation. So to fix the problem, we initially thought that we could just do genome engineering and this would solve the problem. This turned out not to be true completely. Then we went about trying to engineer the medium such that you would switch from glycolysis to oxidative phosphorylation, and then ultimately to do further engineering and actually do niche engineering. So the transcription factors we found, we tested about 7 of them, and we found that 3 of them actually allow you to improve the differentiation. So what we did is actually introduced the transcription factors in the safe harbor locus under an inducible CRISPR-Cas system. And shown here is that we have a TET-on system that drives the transcription factors that is present in one locus in the genome of the cells. So the transcription factors we overexpressed were HNF1 alpha, FOXA3, and PROX1. You can see here that the overexpression shown for each here, HC3X, which are the 3 transcription factors, is actually in the range of primary hepatocytes cultured for 12 hours or primary hepatocytes freshly isolated, except that HNF1 is a bit higher than what we see in these cell types. This improves albumin expression, this improves CYP3A4 expression, but you will notice that CYP3A4 expression is still lower than in primary hepatocytes. And is also at the functional level, so albumin secretion is improved, BFC metabolization is improved some, but we're still a long way away from primary hepatocytes. So then Ruben Bohn in the lab actually started looking at whether we actually could change, whether we could understand the cellular metabolism. And he did 13C labeling of pyruvate, lactate, and alanine to assess which of these actually are incorporated into glycolysis and TCA cycle. And he also measured the disappearance of different carbon sources from the culture medium. And these studies made us to conclude that low amino acids in culture medium are responsible for poor lack of gluconeogenesis and drug biotransformation. So what we showed is that if you increased amino acids, shown here as amino acid 3, and shown here in gray, is that if you do this, so the 3x modification plus the improved medium, that we now get gluconeogenesis just like primary hepatocytes, and that we start improving oxygen consumption compared to HLC without the improved medium. This translates in an increased expression of CYP3A4, which you can see here, that if you use this medium here, that we actually get CYP3A4 expression that is very close to primary hepatocytes. And interestingly, it also translates in the ability of these HC3X or HC6X, which is the other line that we made, cultured in the complete medium, to now be able to metabolize midazolam to the same extent as primary hepatocytes. You will also note that not all CYP enzymes are equally functional as in primary hepatocytes. For instance, CYP2D6 is not really metabolizing dextrometorphan, but we can metabolize tolbutamide and phenacetin to some extent, suggesting that the cytochrome function is improved in these cells. So in conclusions, we started out with immature, poorly functioning stem cell-derived hepatocyte progeny, and by doing 2 things, which is CRISPR engineering and nutrient engineering, we can change the cell type of these cells to produce glucose and efficiently biotransform at least some drugs. And I will come back to toxicology studies down the line. However, these cells are still not quite the same as primary hepatocytes, as I just showed you. And also the transcriptome studies show that, and that was published in this paper in Nature Communications. So the next question was, what if we try to co-culture these cells with the other cells present in the liver sinusoidal niche? And so everybody now knows that 3D culture better mimics the organ in vivo. And so how to mimic the liver niche? We use the 3rd engineering approach, namely bioengineering. So to do that, we have to make 3 cell types aside from hepatocyte-like cells. So we need to make stem cell-derived stellate-like cells. And this was published in 2018 with now a very detailed protocol in 2021, where we take iPSCs and use different growth factors to drive them to a cell that has stellate cell-like features. I don't think we can truly call them stellate cells, just like you cannot call most cells in vitro similar to in vivo. Nevertheless, what we showed is that these cells can be generated. They store retinol, and actually, if you stimulate the cells with TGF-beta, for instance, you can show that they become activated. And if you put them together in spheroids, together with, for instance, HEPA-RG cells, we can show that the cells actually make spheroids and can actually cause fibrosis if you treat these spheroids with pro-fibrosis causing agents. So it looks like you can make a stellate cell that is probably better than a classical stellate cell that you harvest from the human liver and you maintain for multiple passages in vitro, even though also our system, if you grow them long-term in vitro, you get a progressive activation of the cell phenotype. The second cell we generated is stem cell-derived endothelial cells, and then we tried to make them a bit more liver sinusoidal, and it was also published just very recently. To make endothelial cells, it's very simple what we did. We actually put the transcription factor ETV2 again in this AAVS1 locus, either in ES cells or iPS cells, and we just doxycycline for 8 days. The whole population of embryonic stem cells or induced pluripotent stem cells changes over in endothelial cells that actually make beautiful vascular tubes in vitro. Now, these cells are classical run-of-the-mill, probably HUVEC-like endothelial cells that don't express any typical features of liver sinusoidal endothelial cells. So by doing RNA sequence data mining, we identified a top list of transcription factor candidates and actually took the top one and put it together with ETV2 into this AAVS1 locus. And so it's actually a transcription factor called SPY1. They're also PU.1. So if you now overexpress ETV2 and SPY1 together, again, just using doxycycline from day 0 to day 8 to 12, we get cells that start having some features of liver sinusoidal endothelial cells. And shown here, for instance, is that they can uptake IgG, or they can uptake beads via multiple scavenging receptors on the cell membrane. And shown on the left-hand side are actually on the top panels are mouse cells, and in the bottom panels are the human cells. What we could not yet show is that we do not see the typical fenestrations, but these cells are all maintained in 2D culture. So we can make cells with endothelial cell features for sure, and we can push them a little bit to get liver sinusoidal endothelial cells. But again, they are not fully liver sinusoidal endothelium. The last cell we made were macrophages, and this is the protocol we used initially with the idea to make microglia, but you actually make macrophages on the way to microglia. And actually, we can get a population that's 100% CX3CR1 positive and CD14 positive. And actually, these cells, which are called MC or macrophages, differ quite a bit from macrophages in vivo, but seem to cluster with all the tissue-resident macrophages in different organs. So then to try to make a 3D system, what we did is we tried a number of extracellular matrix molecules like collagen, fibrinogen, fibronectin, and so forth, but the cells seem to degrade these matrices quite easily. So Manoj Kumar in the lab actually developed a polyethylene glycol polymer-based extracellular matrix hydrogel. So this is a PEG polymer to which we can link at the ends peptides that we represent extracellular matrix molecules or cell adhesion molecules present in the liver. And then you can actually link all these polymers together using peptides that are slowly degradable by MMPs. So once we had all the combinations that we had here, we actually had 15,000 different combinations that we had to test. And actually what we did is we made a design of experiment, still needed to test 216 combinations of different stiffness, different degradability, and different combinations and permutations of extracellular matrix and cell adhesion molecules. So we first started to put hepatoblasts into the culture, into this hydrogel. So what we do is we let the hepatocytes differentiate until day 8, harvest them, and put them as single-cell suspensions in this polymer, and then let them differentiate. Initially, we let them differentiate till day 20. When we do this, we can actually then rank order the different hydrogels and try to figure out which population or which hydrogel seems to be closely linked to primary hepatocyte phenotype. And this is RT-qPCR for a limited number of genes, but that was then confirmed for a much larger complement of genes, as well as CYP3A4 function. And so we found this hydrogel, and it doesn't matter too much what the letters are, but MP2, DG2, PP15, that seemed to be most closely making cells that were more closely related to primary hepatocytes. And here, this black line is the 2D control. So you can see that you can significantly improve the maturation. We did this in baseline medium, as well as in this optimized medium. And each time, this hydrogel was the one that most closely linked to primary hepatocytes. If you then look what happens in these hydrogel, you see assemblies of small spheroids of what looks like hepatocytes, which is shown here also to stain positive for albumin, HNF4 alpha. Some cells are CYP3A4 positive, but some cells are still AFP positive, but CYP3A4 negative. The cells express NTCP and occludin. And interestingly, so if you go from 2D to 3D, CYP3A4 function improves, and now you get in the region of primary hepatocytes, but it's nicely also inducible by rifampicin. We're currently in the setting of a European project, UTOXWISC, actually looking at drug metabolization of this model compared to the other models, just like we did for the 2D culture, but this data is still pending. We then hypothesized that as hepatocytes and non-parenchymal cells are present in the same location in vivo, they actually should be supported by the same, what we call HepMat hydrogel in vitro. So we first tested whether the 3 cell populations individually could be maintained in the culture system. And actually shown for endothelium is that indeed the cells remain V-adherent positive. Interestingly, if you look at some of the LSEC markers, they might be slightly higher if you put them in this specific hydrogel, and they continue to be able to make tubes until about 15 to 17 days in these hydrogels. But this is actually 4 weeks in the hydrogel. We asked the same question for stellate cells, thinking that the stellate cells might deactivate because you put them in a soft microenvironment. And so what we show is that indeed there is quite a bit of vitamin A uptake and neutral lipid staining, quite a bit more than in 2D, suggesting that they are indeed less activated. This is shown by a decrease in collagen 1 and collagen 5, and an increase in the rate limiting enzyme for these fat droplet formations, LRAT. And interestingly, if you then treat the cells with TGF-beta, these cells that actually were less activated, you can reactivate it, shown by increased collagen 1, decreased LRAT, and also collagen secretion. So you can maintain stellate cells again for 4 weeks or more. In this hydrogel, they deactivate but become reactivatable. And the final population we tested were those macrophages. The macrophages remain CD45 and CD11B for more than 4 weeks in vitro. Interestingly, again, maybe there is a slight increase in some typical Kupffer cell markers, but still these are very low, and these are probably much lower than what a primary Kupffer cell would look like. But also here, if we now take these cultures and expose them to LPS, the Kupffer cells remain activatable because we can induce IL-6, IL-1 alpha, and TNF alpha. So these studies showed us that the same hydrogel can actually not only support hepatocytes, but also seems to be able to support the other 3 cell types, although granted, maybe it could be further optimized to improve this differentiation, but that's work that's still ongoing. But knowing that they can be that they each individually can be cultured, we then performed co-culture experiments. So we took the hepatoblasts at day 8, stellate cells at day 12, endothelial cells at day 8, and macrophages at day 16, and then co-embedded them in these hydrogels and let them go for 30 days in vitro to ask what happens to these cells if you co-culture them together. So what you can see is again that you get multiple different clusters of cells that spontaneously make structures in these hydrogels with areas that really look like hepatocytes, areas that look like endothelial cells, and then less defined structures. And we did a Milan analysis. So we actually stained all of these all of these sections with 12 different antibodies to try to understand what the representation of the different cells is in this co-culture system. And this is what we found. So the number of cells that we put in is 1 hepatocyte, a half a stellate cell, a half an endothelial cell, and a quarter of a macrophage. So 4, 2, 2, and 1. What we get out after 30 days is a number of endodermal cells, which are shown here as hepatocyte intermediate, hepatocyte mature, hepatocyte mesenchymal, progenitor, and cholangiocytes. Also endothelial cells in a relatively small number, but now with quite significant number of macrophages and a quite significant number of mesenchymal cells. You will notice something that we call blank. And these were called blank because our criteria to call a cell a specific cell type was very, very tight. So if we weren't sure that any of the combinations of 12 antibodies gave us a clear answer, we called them blank. So that's why really we could only characterize about 65% of the cells, of which then 50% are hepatocytes, 25%, well, 20% are macrophages and 20% are mesenchymal, and a smaller fraction is endothelial. But so compared to the studies that I showed you for spontaneous IPS differentiation, we have a significantly larger number of macrophages, but we also start seeing endothelial cells. Single nuclear RNA sequencing is currently ongoing to try to understand the effect of co-culture of these different cell types together on the differentiation of the different cell types. So that data is still pending. Interestingly, if we now look at this co-culture system compared to a monoculture system in 3D, and again ask how do these hepatocyte-like cells function, you can show here that we already showed that monoculture to co-culture, that 2D culture to monoculture actually improved hepatocyte CYP3A4 function. It further improves in the co-culture system and it remains rifampicin inducible. So this would suggest that co-culture of the different cells may further enhance hepatocyte maturation. And indeed, if you look at the bottom here, 15.6%, or it's probably 25% of the cells in these co-cultures now are CYP3A4 positive, but AFP negative. And so these are really mature hepatocytes. But single nuclear RNA sequencing will obviously need to be done to further demonstrate how close this population gets to primary hepatocytes. So then to start testing the other components of the co-culture system, we did studies where we tried to model NASH. So essentially we took the HepMat co-culture systems or the HepMat monoculture systems and exposed them to oleic acid for 3 and for 7 days, and asked whether we could see liver steatosis, whether we could see fibrosis and inflammation, and whether the co-culture is better than any of the monocultures. So we can obviously show that you get massive uptake of lipids in different cell populations, mainly the hepatocytes in the culture system. If you then, and so we did qRT-PCR, but also these are all ELISAs. If you go to the right-hand side, so these are the hepatocytes on their own in 3D culture, the stellate cells on their own in 3D culture, and macrophages on their own in 3D culture. And you can see if you look at pro-collagen production in any of these cell populations, we don't really see pro-collagen secretion in the supernatant, even though these cells are really the cells that should make it. We also notice that we actually do not see induction of inflammatory cytokines in any of the individual cell populations. However, in the co-culture system, we see that 3 days and 7 days after adding these lipids to the culture system, we get significant induction of secreted collagen. Plus, we also get induction of TNF-alpha, IL-1 alpha, and IL-6, and actually a massive amount of IL-6 in the co-culture, which you don't see in the monoculture. So something in the monoculture must be happening, obviously, and some cells must be responding to triggers that then become probably accentuated and trigger other cells in the culture system. So which cell in the system produces IL-6, IL-1, TNF, or even collagen is not known. And so we're now doing cultures where we remove one cell by one cell to try to understand who is responsible for what we actually see here. This pro-fibrotic phenotype can also be shown, for instance, here, where you can see presence of alpha SMA in the activated cells. And you can see here that the hepatocytes get lipid-filled droplets, which is consistent with the oleic acid stain that you can see up here. Interestingly, when we look at these cultures and then test whether this model can be used to try to test drugs that would counteract NASH, and we used obeticholic acid. Obeticholic acid is one of the only 2 drugs that is now more or less approved by FDA as a medication against non-alcoholic steatohepatitis. And then we tested a second drug, elafibranor, which was very, very suggestive to work in preclinical studies, but also in phase 2 clinical studies. But unfortunately, in phase 3 clinical studies, this failed. So if we now treat with oleic acid with or without obeticholic acid or with or without elafibranor, what do we see related to collagen production and inflammatory cytokine production? And we did this in 2 different populations. We did it in a human iPSC model and a human embryonic stem cell model. What you can see for pro-collagen is that actually obeticholic acid seems to improve pro-collagen secretion in both the iPS model and the human embryonic stem cell model, which is less pronounced for elafibranor. And if we look at the effect of the elafibranor on production of TNF-alpha, IL-1 alpha, and IL-6, you can see that both drugs might actually decrease the induction of these cytokines. Obviously, these are only 2 drugs, and so we're currently in the process of testing a library, a blinded library provided to us by a biotech company to try to figure out how well the model would allow you to identify potential anti-NASH drugs. So I already told you that we actually, if we do genome engineering and nutrient engineering, that we can get some ways and the hepatocytes become better. I showed you now that if we do the niche engineering and actually put the cells in 3D, that we also have further improvement in hepatocyte function. We might be able to get cells with improved LSEC, stellate cell, and macrophage phenotypes, although single nuclear RNA sequencing still needs to be done to try to demonstrate how close to the real thing we get. And it can maybe be used to model more complex liver diseases like NASH, cirrhosis, or other diseases. And then obviously you will ask, can this be used to evaluate toxins? So for that, we actually optimized both the 2D system and the 3D system to high throughputs. And so we have a fully automated system wherein we can actually have the robot plate undifferentiated iPS cells in either 96 or 384 well plates, and then do the hepatocyte differentiation fully automatic for till day 40 or day 50, obviously then dispense drugs and have automated imaging at the end to try to understand what's going on. So it can be done in 96 and 384 well-played format, so you can go through a reasonable number of drugs, although the assay is obviously still quite expensive because creating hepatocytes are quite expensive. For that, we're actually currently looking into whether we can remove some of the very expensive cytokines by small molecules, again, using the same robotic system. So when we do this and we take iPS or embryonic stem cells and put them through the 40-day differentiation and then expose them to different chemicals, what we are able to show is, again, that if we use the A-glide, so the optimized medium, and this is again the 6X, not the 3X, but so these are genome engineered cells evaluated on day 40 or day 50, looking at toxicity by APAP, amiodarone, and rotenone, even though APAP was not identified in hepatocyte-like cells that weren't genome engineered and weren't medium engineered, and amiodarone was not identified, nor rotenone, you can see in blue and red that after 40 or 50 days, the cells have a phenotype now that looks very much like primary hepatocytes. And this is true for embryonic stem cells and an iPS line. We also tested the IMI training compound list, which contains all of these drugs listed here. So these are non-toxicants to the liver, and these are controlled toxicants to the liver. And again, in black is hepatocyte-like cells without genome engineering and without medium engineering. The gray is without genome engineering, but medium engineering. And then purple and blue are genome engineering and medium engineering. And then to find out whether genome engineering is important, we actually removed the doxycycline on day 30 and let them in the optimized medium until day 40. And what you can see is that each time underneath the stippled line that these drugs are toxic, such that, and that this is actually significantly more, that the purple and the blue bar are significantly more sensitive to the toxins than the black bar shown here or the gray bar. And also, interestingly, the light blue bar, which is the dark blue bar minus the transcription factors for the last 3 days. In another way to show this is if we take a cutoff of 10 Cmax, we can show that we can pick up almost all known toxicants except one in the 3X or the 6XA glycine system. But if you remove this overexpression of the transcription factors, we actually lose the sensitivity. These are HepG2s as control, and we did not do ourselves primary hepatocytes, but in the original paper where this was described, the primary hepatocytes actually are more or less identical to what we show here for iPS-derived hepatocyte-like cells. We then did an initial benchmarking study of the transcriptomics done for one drug, because the initial transcriptomics that we did, and this was done as part of a Marie Curie project called IN3. So we exposed cells to diclofenac and did tempo-seq analysis of transcriptome changes 24 and 48 hours after exposure of hepatocyte-like cells to diclofenac, and then looked at the transcriptome studies that have been published for primary hepatocytes, HepG2s, HepRG, and rat hepatocytes. And so SBAT2, 3X are actually the hepatocyte-like cells created by addition of the 3 transcription factors, but also amino acid glycines. And you have to pay attention to the concentration. So primary hepatocytes exposed to 400 micromolar actually induced about 1,200 genes that were differentially expressed. But the next ones at 75 micromolar, 37.5 micromolar, or 7.5 micromolar, 24-hour exposure of the HLCs actually give us the induction of about 600 genes that are aberrantly expressed. If you look at HepG2s and HepRG at much higher concentrations, here the induction of transcriptional changes is significantly lower than what we see here. So we would dare to say that for diclofenac, this model is actually quite okay to identify transcriptional and therefore mechanistic insights in how drugs might be toxic. To extend this further, using in the context of EU-Tox risk, and also that will continue in Risk Hunter 3, we also started to put mode of action reporters in pluripotent stem cells, so we knock in color genes at the end of the endogenous gene in the genome as a fusion protein, much alike what Bob van der Water actually showed with HepG2 reporter lines. So essentially, we use CRISPR-Cas to knock in GFP or another color with a linker linked to the protein that will be expressed. And the first example of this is actually HMOX1, which it represents oxidative stress sensor. And so if you take human-induced pluripotent stem cells and expose them to oxidative stress molecule, CDDO, you can show that 24 hours after the exposure, most of the cells are green, and we can actually do dose-dependent and time-dependent lifetime imaging of iPS exposed to these toxicants. These cells were then differentiated into 4 different cell types: cardiomyocytes, proximal tubular epithelial cells, hepatocytes, and neurons. And again, most of these should actually probably, like hepatocytes, have like in between. But what we can show is that there is actually different sensitivity between the different cell types and also a different time course phenomenon. So the strength therefore of iPSC is that you can actually do toxicology across different tissues using the same initial cell population. And the second major advantage is that you can actually relatively easily do genome editing to incorporate these reporters in the cells. Then finally, we're in the process now of optimizing and fully automating the 3D culture system. And this is just a gel drop without any cells. We then made gel drops with HepG2s till day 40. And then these are the 3xAG cells for day 40. And we're characterizing them now. And we will move from just hepatocytes to actually the co-culture system once this proves to be satisfactory, because then this will allow us to also do toxicology in more high throughput in an automated fashion, but in a much more complex model of the liver. So in conclusion, the stem cell models I showed you may form an alternative, but for sure an adjunct to animal models for disease modeling and drug discovery and toxicity studies. The strengths are obviously that they're all human models. They can recapitulate a number of aspects of a tissue and organ and can capture certain disease genotypes and phenotypes, but likely also the phenotype of toxicology. But hopefully I showed you that we are making significant progress in inducing improved maturation, but we are 100% for sure that they're still not quite as mature as they are in an in vivo organism. And what I showed you in the 3D systems is that we get multiple cells together, but it's definitely not quite the same as what a liver would look in vitro, in vivo. It isn't clear whether that's necessary to get full functionality, but it's still something that could be looked into. So we need to further optimize culture conditions, including ECM, nutrients, and ligand engineering. The co-maturation, I think, of the different germ layer cells probably will improve differentiation, but we need to actually get the single nuclear RNA sequencing data set analyzed. As I mentioned, whether the tissue organization is necessary and you need to do bioprinting is an open question. And obviously there is no perfusion and hypoxia might still be a major problem. So microfluidics would be interesting. And then you could also move it in more complex systems like a multi-organ on a chip. So obviously this work was not done by me, but by the many people in my laboratory. Laura was instrumental at the initial genome engineering. Manoj has really done all the 3D bioengineering. Rodrigo, Jokicin are people that are involved in the robotization. Ruben was the author of the Nature Communications paper, and these are the additional doctoral students that have contributed to it. We got funding from multiple European organizations, eBISC, HemiBio, EU ToxRisk, IN3, and RiskHunter, local Belgian organizations, charities, as well as companies. And these were all the collaborators on this project. I will stop here and I can take questions. Thank you very much.
Thank you indeed very much for that. It's really comprehensive and data-driven presentation. Lots of information there, and based on the questions, I think people have gotten a lot out of it. So we'll have time for a couple of questions. The first was, do you routinely use atmospheric levels of oxygen for culturing hepatocyte-like cells?
So these are done in a normal ambient oxygen incubator. So we don't use hypoxia. In 2D, it might still be okay, but 3D, I think you would really become very problematic because hypoxia is probably a problem in the 3D culture system. And it would be interesting to look at different gradients of oxygen to see if you can get zonation to be mimicked. But again, the single cell and the single nuclear RNA sequencing may actually provide us some answers there.
Okay, thanks. Can you recommend guidelines for how many different hiPSC lines should be compared to indicate if variation among individuals in the population is a factor that needs to be addressed?
So we have currently looked at 4 different hiPSC lines. I know that's not many, it's not 20, 30, 40, but we do see some differences. So for instance, we have used an iPS line that we purchased from Sigma, and that actually behaves quite similar to human H9 embryonic stem cells. But then we have used other iPS lines like the ESPA2, which is from by the IN3 consortium, and they behave a little bit less good. So actually the drug metabolization is a little bit less good. And we have an BJ1 iPS line, which is also similar to one of the first ones that Shinya Yamanaka published. It also has some variation. I think we haven't really looked at enough of them to be able to say that this truly represents the human variation. Plus, ideally, you would get cells from people that have been very well characterized, that the hepatocytes have been very well characterized, to try to understand whether what we create from iPS, recreates what hepatocytes would look like. So you would really need to take blood cells or fibroblasts or hepatocytes, reprogram them from a patient with a certain genotype, phenotype characteristics, and then see whether you can mimic this in vitro. But that would be a huge undertaking. I think that would have to be a huge consortium doing that.
Maybe we'll try one more. Have you looked at flavin-containing monooxygenases in addition to P450 in your cell systems?
So we have data together with the University of Valencia, and so there we do see increased functionality in the improved cells. So if you do genome engineering and metabolic engineering, these functionalities also improve. But again, they're not exactly quite the same yet as primary hepatocytes. As I mentioned, the 3D system under UTOX-WISP, we're actually now doing drug metabolization and for both phase 1 and phase 2. Comparisons between primary hepatocytes, HepG2s in 2D, 3D, HepRG, and so forth, but that data is still pending. And the co-cultures, we actually haven't looked at it yet.
Okay, thank you. I'm sorry we didn't get to all of your questions, but we need to move on to the next presenter now. So thank you so much, Dr. Verfaillie. Really excellent talk to add to the level of knowledge of all of our attendees. We will now move on to Anne-Marie Vinggard. Please go ahead.
Here we are, I think. Do you see my presentation?
Yes.
Thank you, Christy, for the introduction. I'm happy to present some of the work we have been working on for the last 5 years or so, dealing with predicting developmental toxicity with human-induced pluripotent stem cells. So a human being starts with a fertilized egg that undergoes several rounds of cell divisions to form the blastocyst on day 4. And cells in the blastocyst will form the inner cell mass, which in a first step form the 3 germ layers and becomes an elongated structure. And here we can see the beginning of the heart development with the mesodermal cells that migrate to form the primary and secondary heart field, which will later form the complex structure of the heart. And on day 22, the heart beats for the first time. And until the 9th week, all organs are formed and the embryo becomes the fetus, which is growing constantly until birth. Developmental toxicity is defined as adverse effects of chemicals or drugs that are induced during pregnancy or as a result of parental exposure. And one of the best examples we've got is the drug thalidomide, which was prescribed for nausea, among other things, in pregnant women in the 1960s, and caused a disaster as around 10,000 children were affected, and half of them died and half were born with lacking limbs or other adverse effects. And thalidomide was not detected as a developmental toxicant in the preclinical studies using rodents, and therefore the rabbit was later introduced as a model for developmental toxicity. But today, the wish is to test large numbers of chemicals in a short time for developmental toxicity, and this requires in vitro tests that can be run in a high-throughput format. So this slide illustrates some of the current in vitro and ex vivo approaches that have been developed for developmental toxicity testing. You can use either embryos from either the rat or the zebrafish or other animals, or you can use embryonic stem cells derived from either a mouse or a human being, or the latter test here based on the mouse embryonic stem cells. The embryonic stem cell test is one of the more widely used models. But also a reporter gene assay called the ReproGlow has been developed, which is based on testing for adverse effects on the Wnt signaling pathway. But to overcome the challenges of the species-specific outcomes and ethical issues with using real embryos, the opportunity has arisen to use induced pluripotent stem cells that are of human origin and for which there are no ethical issues. So here's illustrated how embryonic stem cells are formed from the fertilized egg and require an embryo to be formed. But induced pluripotent stem cells can be derived from adult blood or skin cells and can be differentiated into various organs of the body, such as the heart. And we have taken advantage of embryonic bodies made from human-induced pluripotent stem cells. Embryonic bodies are 3D clusters of stem cells that resemble the blastocyst, which mimics the very early stage of an embryo. And to the right is here seen a photo of an embryonic body, and it can differentiate into various germ layers. And here we form embryonic bodies in conical shaped culture wells and differentiate them into heart tissue, which is the very first organ to be formed in the body. And we have employed an 8-day protocol, which was originally developed by the Fraunhofer Institute and further optimized by our former PhD student, Karim Lauske. And the cells, they are seeded on day -1, and embryonic bodies are exposed to various medias each day except for the weekend. And the embryonic bodies are exposed to chemicals on the days indicated by the red arrowheads at day 1, 2, and 3, and 6. And the composition of the media, the various media, is very complex, and detailed information can be found in the paper mentioned below here. And at day 7 of the assay, the differentiated embryonic bodies look like this in a microscope. They are beating like real cardiomyocytes, which is fascinating to look at. So we have characterized the embryonic bodies by analyzing various marker genes for mesoderm formation or early or late cardiomyocyte differentiation. And we found that the mesoderm markers illustrated by the green lines here, they peak around day 1, whereas the early cardiomyocyte markers illustrated by the blue lines peak around day 2, whereas the late cardiomyocyte markers peak around day 6 or 7. And this was confirmed by immunohistochemistry, and simultaneously the pluripotency markers OCT3, 4, nano-G, or SOX2 decreased as they should during the differentiation process. So we started out by using a Bioni 10 clone that was developed by the research-based company Bioneer here in Denmark. But we wanted to include more clones, and selected the 2 commercial clones, IMR91 and IMR94 from Stem Cell Technologies. And we showed that they were all able to form beating cardiomyocytes. So we also characterized the other HIPS clones, and we found out that they gave almost similar results as the Bioni cell line, except that the IMR91 did not lose pluripotency as efficiently as the other clones, as can be seen here to the right on the nano-G and the SOX2 expression, which is higher in the IMR94 clone. Then we started testing chemicals in our assay, and we tested 3 chemicals, salidomide, which is considered the golden standard as a developmental toxicant, valproic acid, which is more a neurodevelopmental toxicant, and epoxyconazole, which is a fungicide that we have previously shown is a developmental toxicant in rodent studies. And we found out that our assay detected salidomide and epoxyconazole as positive compounds in our assay, whereas valproic acid was negative. And the HIPS line differed to a minor extent. I think overall we got more or less or very similar results, except that for the IMR94 clone, it was not possible to have a significant response of thalidomide at these concentrations in that clone. So therefore, we have suggested to use a tiered approach for chemical testing. And we suggest to test first chemicals in the Bioni 10 cell line, and if it's a positive, to determine IC50 value in that cell line. If it's a negative, then we suggest to test in the IMR91 cell line, to detect if the chemical is positive or negative. And we used that approach for testing 3 fluorinated chemicals, PFOS, PFOA, and GenX. And PFOS and PFOA are well-known developmental toxicants. They are in human. They are associated with reduced birth weight of newborns in epidemiological studies. And GenX is a newer fluorinated chemical that is replacing the old well-known fluorinated chemicals now. So we tested these 3 compounds and found that both PFOS and PFOA was positive in our assay, whereas GenX was negative. We tested the GenX again in the IMR91 clone and found a weak positive response in that assay. Then we asked ourselves if we can improve the pluripotent assay, as we call it. The assay is relatively labor intensive, and it requires a lot of work to count the beating cardiomyocytes at day 7 of the assay. So we wanted to introduce a genetic reporter into our HIPS line in order to measure luminescence intensity instead of counting beating cardiomyocytes. So this slide here is a schematic presentation of our genetic engineering strategy that was done by Andreas Treschow and collaborators at Bioneer. We introduced a nano-look luciferase reporter gene into the locus of NKX2.5 of our HIPS line, and this enabled us to measure luminescence intensities instead of counting beating cardiomyocytes. And we retrieved 2 clones of this reporter cell line and showed that they differentiated into cardiomyocytes with the same efficiency as the wild-type cell line. And we subjected the clones to detailed quality control assessments to verify their pluripotency and genetic stability. And to the left is shown the sequencing results of the mutated areas of the 2 clones. And to the right is shown the karyotype that was normal in both cell lines. We also analyzed the expression of pluripotency markers in the 2 clonal cell lines by immunohistochemistry, and both lines expressed the pluripotency markers, suggesting that the cells retained their pluripotency during the genetic engineering. And in order to get a quantitative measure of the proportion of pluripotent cells in the cell populations, we performed flow cytometry and showed that both clones expressed pluripotency markers to a very high degree. So moreover, to confirm the ability of the cells to form derivatives of the 3 germ layers, we subjected them to differentiation protocols into ectoderm, mesoderm, and endoderm. And flow cytometry analysis of the population showed that a high proportion of the cells expressed markers for the respective germ layers. So in conclusions, we think we have confirmed that the 2 clones had retained their pluripotency and were able to differentiate into the various germ layers. So then we measured luminescence during the course of differentiation and found a strong and significant increase over time in both clones of the reporter cell line. And this correlated well with the increased expression of NKX2.5 during the course of differentiation. And moreover, NKX2.5 was expressed at similar levels in the 2 cell lines. And together, these data indicate that luminescence intensity mirror the expression of NKX 2.5 and was indicative of cardiomyocyte differentiation. So it was important to prove that the differentiation into cardiomyocyte was not hampered by the genetic engineering. And we exposed the cells here to thalidomide and compared the readouts, beating cardiomyocytes indicated by the blue columns and the luminescence indicated by the red column. And we found that thalidomide decreased luminescence intensity significantly and to a higher degree than the beat score. And therefore we decided to repeat the experiment and lower the concentrations of thalidomide even more. And the results are shown here. We found that at concentration of 0.3 micromolar thalidomide, the luminescence intensity was decreased, and we did not see any effect on the beat score at that low concentration. And this was repeated for the other clone. So at least for thalidomide, the luminescence assay that we have termed the pluripotent assay seems to be more sensitive than the pluripotent assay. And then we also tested valproic acid, which was also negative in the luminescence assay, and we found that epoxyconazole also gave rise to a sensitive response in the luminescence assay. So at the moment, we are expanding our database and we are testing more chemicals in order to more detailed define the applicability domain of our model. So in conclusion, the human-induced pluripotent stem cells seem to be a promising tool for developmental toxicity testing. Our pluripotent assay detected thalidomide, epoxyconazole, PFOS, PFOA, and GNX as positive compounds, whereas valproic acid was found to be negative. And we have provided a proof of concept that a genetically engineered HIPS line with a luciferase in the locus of NKX 2.5 could be developed. A reporter cell assay that we have termed pluripotent assay. And we found no difference in cardiomyocyte differentiation between the pluripotent and pluripotent assay. And we think that the pluripotent has got the potential to be a good tool for future chemical testing. But we need to expand our database and to test more chemicals to define more exactly the applicability domain of our assay. So, and finally, I would like to thank the students that have been working on this in our lab at DTU Food, Karin Lauske, Andreas Treschow, and Niklas Davidsen. Also our collaborators at Bioneer, Bjørn Holst and Mikkel Rasmussen, and the people at the Fraunhofer Institute, Julia Neubauer and Ina Meiser, and my co-supervisor at DTU Bioengineering, Jenny Emneus. So with these words, thanks for your attention.
Thank you very much, Professor Vinggard. That was a really nice talk, and lots of good information to lead to some testing methods. We have a few questions, so I'll just start right off. The first one is wondering about whether it's necessary to differentiate or whether it makes a difference as to whether the cells are derived from males or females.
Yes. As far as I remember, we have used these 3 clones, and 2 are derived from the one gender, and the third is from the the other gender, and we haven't seen any difference at the moment. But of course, our database is not that extensive at the moment. Right. But we haven't found any sign that either age or gender or sex is important for the differentiation process.
Okay. What was the proportion of different cardiac cell types in your organoids? So atrial, fibroblast, ventral, etc. Did you analyze that?
We have not defined exactly which cell types are present in our cardiac 3D cultures. We have made a study where we have compared the expression of markers in our embryonic bodies based on hips with a peak embryonic cardiac markers. So we have tested a variety of peak embryos at various stages of development, and compared the expression of markers in the pig embryos with markers in our human embryonic bodies. And we found many similarities, but also some differences. And I think the overall conclusion, our 3D model is a little more to the immature side than to the mature side, if you can put it that way.
Yeah, that makes sense. So the next question, it has to do with the nCardia technology. I'm not sure if you're familiar with that assay, but they're asking about comparing the advantage of the pluribeat to the nCardia.
Oh, the nCardia. I'm not. Is that another? I'm not familiar with that assay or that technology. How do you spell that?
They spell it NCARDI.
No, nCardia technology.
Yeah.
It may, I don't know, I guess it may be a technology they are using in many pharmaceutical companies where they are where the assay is more or less automated. But I think in those applications, or in many of those applications, they are using 2D cultures of cardiomyocytes and not the 3D cultures as we do. So I think we have an advantage in using 3D cultures of cardiomyocytes as they become more physiologically relevant.
Okay. There's a question about compounds that specifically target neurons, and whether the assay would pick those up, or whether there's additional need.
Yes, we found that the valproic acid, which is considered a developmental toxicant, was negative in our assay. And we think that there are some indications that it maybe is more neurodevelopmental toxicant. So it affects the brain. And this may be the reason why we don't see a positive response with this compound in our assay.
Okay.
So
you actually answered two questions with one answer because the question was about valproic acid. But this other question is about thalidomide and whether it has a specific mode of action on cardiotoxicity, which, it's more related to vascular.
Yeah, actually, we are not using this assay because we are really interested in detecting cardiac toxicity. Our aim is to use the assay to test for developmental toxicity or embryo toxicity. I think the mechanism of action of thalidomide is still under investigation, but it's not definitely not specific for cardiac toxicity. It's an angiogenesis inhibitor, but it has also a variety of other mechanisms of action. So yeah, which is still studied. Yeah.
So I think that there's a little more information on nCardia here. It's a company that produces human cardiomyocytes.
Okay,
So,
yeah. Sorry, I don't know this company. Yeah, I'll look into that.
So there was, I think we'll have time maybe for one last question. There was a question that I wanted to return to. It was actually asked during Catherine's presentation, but I think it's relevant for both of you, and it has to do with, it asks about the ideal proportion of different cell types for toxicity testing, and would it be different for different challenges? So for acute toxicity or ROS production, immune-related injury, those are just examples. But I think for both of you, maybe if you could talk a little bit about targeting, which we've talked a little bit about just now, Anne-Marie, but targeting the type of toxicity, how important is that versus a model that can assess different kinds of toxicity? Or do you need multiple cell types to do that? Would you, anything to say about that?
Catherine, do you have a good answer to that?
I don't know if I have a good answer to that. I may try an answer. So I think what I try to show is that if you have a single cell that is quite potent, so for toxicology, obviously in the liver, hepatocytes are the cells that die, and people, suffer from losing hepatocyte function. But what I try to show is that if you expose the different cell types to excess lipids, that if you give it to a single cell at a time, you don't see much. But if you give it to the complex of hepatocytes plus the other 3 or 4 cells, that you start seeing what actually might be happening in vivo. Whether that's really what's happening in vivo, we don't know. But at least you start seeing inflammation that you wouldn't see if you just give this one trigger to hepatocytes alone, or even macrophages or stellate cells alone. But you start seeing the interplay between the cells. How many of each do you need? So we obviously didn't quite mimic in vivo liver, and they're not in the same physical assembly that they are in vivo. So, how important those things are, I don't really have an answer for. But I think more complex mimics of the human tissue may give you more informed answers than if you do single cells at a time. But it becomes much more difficult to interpret. So I don't know for you, Anne-Marie, if that's also the same things that you have in mind.
I think it sounds reasonable. I haven't got a better answer to that. I think it's really a great opportunity with the hips that you can differentiate into so many different organs and cell types. And I think that could be a huge advantage. And I think it's also an advantage that you can maybe mimic better individual differences in the population. You can use, let's say, 10 hips clones for toxicity testing. Then it may cover a good broad picture of and represent the human population better than we do now when we're using 2D cultures of cells and
Well, and I think also for developmental toxicity, you're capturing processes that are very early on in development. So, you know, maybe you don't need as many cell types because it's upstream of the development of all those different different organs and tissues.
Because maybe also for developmental toxicity, you know, the early cell types might be easier to make than the true, fully mature, you know, hepatocytes, where the gold standard is, you know, obviously we have very many tools to prove the gold standard, but we still aren't there. So we cannot really make anything that looks very, very close to a true hepatocyte that comes fresh out of the liver. Because from the moment you take it out of the liver, it already changes.
Right.
So it's complicated.
Well, we've reached the end of our time. It's been a really interesting discussion. Thanks to all of you for listening and for your questions. And really, thank you so much to Dr. Verfaillie and Dr. Vinggard for taking the time to present with us and talk to us about your work. Thanks very much.
Most welcome.
Thanks for arranging this, Christy.
Yeah.
Yes.
Thank you.
Thank you.
Bye.
All right. Bye-bye, everybody.